Hello, welcome back to the Computational Genomics Lab. If you're here, that means you've run the Nextflow NF Core pipeline for ATACseq, I think version 2.12, and must be awesome because we have now tons of data, just like we did in the RNA-seq video. NF Core ATACseq pipeline will do a bunch of analyses for us, and so that's what we're going to go take a peek at today. Okay, so we're again on level 8 in class. Here's the class GitHub. You go to lessons, 08 ATACseq pipeline, and ATACseq outputs. Okay, so in here, what I've done is just outlined, you can see it's a very short document, just a few things to think about in the multi-QC. I'm just going to show you where it is, how to think about it, a little bit, real quick, brief overview, but there's more details in here if you really want to get to know it much better. And what are the unique results to Max? I'll kind of touch on a few, but there's a lot. And then there's, of course, the raw data bigwigs. We've started to learn how to make raw data figures in IGB. So we have those in there, and just kind of where stuff is. But we'll now take a quick look at that. Okay, so here's, let me go to where I ran the pipeline. So that was the key thing in the last class, is what you wanted to where you put your files and how you made your sample sheet or design file to those FASTQ files. So I'm already all the way in there, but let's say here's our 08 and lessons here. I had an 00, I ran it in the same class thing, 00 pipeline run, and here were the data input files that you worked on in the last video, and then we have the pipeline run was the output directory I used, and in here we can see BWAs where the bigwigs are going to be. Here, there they are, and then some peak files are also in there, and then the one I want to go over real quick is this multi-QC, and there's a broad peak folder in there. I should note that in the way we ran it, we used broad peaks. You could use narrow peaks, or you could use both. It's in the NF Core documentation, and you can just add more flags to that. I just went with broad peaks for now. For simplicity, that just means it's willing to spread the window of where it says the peak start and stops, which is going to foreshadow the next few classes. Starts and stops are positions of peaks in a sequence of DNA. We'll be doing a lot with those in a second, or in the next lesson. Okay, so let's open this in a web browser. This is our multi-QC report, and this is like number of reads, same stuff we saw before with RNA-seq, and everything looks good. Again, you can just go through these. This is a lot of just the trimming of these things, and let's go down to where there's more like sample information. You can see it just does a ton of work for you. It's amazing. Okay, let's get down here. Okay, so this is a important part of it here called the FRIP score. If you want to find it, you can just find over here in this index. You can get right to it quicker. But the FRIP score tells you the percentage of the reads that were sequenced that were inside of the peaks. And here it's about 10 or 20%, which is not awesome, but also not unheard of. And so that basically means that there's only 10% of the reads come from peaks, and the rest of the reads are sort of scrambled around, which is kind of common in genomic exercises. They're noisy. Here is that matrix plot that we got with RNA-seq that showed us how similar our samples are. And I don't want to give away any results, so I'm actually not going to talk about this on purpose and have you take a look at it and see what you think. We'll do other exercises here, but that is a result in and of itself. Because one of the main goals we're doing with this is to ask, how does doxycycline affect chromatin accessibility? Answer's right there. Okay, so that is all I really wanted to go for over the multi-QC. And you'll see that there's pretty much in here other stuff you wouldn't really need to use. I would just look at the multi-QC, and in the BWA is where most of our stuff is. Same with the RNA-seq pipeline. So we have our bigwigs here that we can drag into IGB. We have DeepSeek tools you probably don't need. Max2 is pretty handy because this ran a whole program called Max, which called all of our peaks. And so we have peak files for every single sample. So I'm going to do that one more time just because this is the most important part of the data you're going to actually need to use is in pipe wherever you ran it, the BWA, merged library, bigwigs are a key one, and then the max2 is a key one. So in RNA-seq we had counts and TPMs. It's kind of similar to this one. We're going to have counts and TPMs in these peak files to know how many counts were in the peaks and some normalization, which we'll go through. But these files are just going to be everywhere that docs affected chromatin, or if it didn't. We don't know yet. Okay, so there's some more detail here on other things you can look in there, but this max2 broad peak folder is where the gold is, so to speak. Okay, that's all I wanted to show for the pipeline outputs. And next we're going to do load in a peak file. And we're using the same strategy we used with the RNA-seq. We did that, of course, pipeline run. We looked at the multi-QC and the output files, the bigwigs. And now we're doing the same thing. Then we imported the counts and TPM files. So in ATACseq, that same exercise is going to be very similar. It's just that we're going to be loading in peak files. But how we interpret those files is going to be different. And that's going to allow us to learn what's called I ranges or G ranges, which are essentially a start and stop of an event in the genome. And we're going to go into that much more detail in the next classes. See you there.